Hello, I'm Dr. BCMR. There are many other mutations that can be apart from the mismatches that are because of replication, majority of them at least. There will be additions or deletions and we onto the bases or anywhere else, and these have to be removed. And there the nucleotide excision repair comes into action. Nucleotide excision repair has a different set of proteins that are involved, and of course the any kind of problem in the base pairing is always, for majority of the times, detected by as a distortion. If there is a distortion, the scanning does happen by proteins called as UVRAB. UVRA is responsible for scanning the DNA for any distortions, which are usually because of some problems with base pairing between the bases, and that is because of many different changes it could be. And UVRAB as a heterodimer, or it's a tetramer with 2 subunits of A and 2 subunits of B, and B is responsible for anytime there is a distortion. B will melt the DNA, that is denature the DNA at that location. So we'll see that there are many bases that are denatured in this location. And once that happens, the UVRA is removed and UVRB is bound, is still in the location, and there is something that happens is recruitment of UVRC. So UVRA, UVRB, and now we have UVRC. UVRC is a nickase. So it's something like it makes a cut here and makes another cut there. So about, if this is the location of the actual damage to the bases, about 8 bases onto the 5' side and about 4 to 5 bases onto the 3' side, it makes a cut. And the whole element, the whole DNA is removed. I hope you see the difference between nucleotide excision repair and the base excision repair. In the base excision repair, only the base that is problematic is removed first, and then the small part is removed, the sugar and the phosphate are removed and then filled up by polymerase and ligase, sealed by ligase. In nucleotide excision repair, we see this whole fragment, a polynucleotide, is removed from the site of injury. A new chance for the DNA polymerase to fill up the gap happens. So once the nick is made, there is another protein called as UVRD, which is the helicase, as we heard before, and it will remove this fragment. We need this protein because there are numerous bases here, say 8 plus say 5, we have about 14, and there are numerous hydrogen bonds, they still be binding. So we need helicase activity to remove this fragment, so that now we have a 3' OH, and that can be taken up by DNA polymerase, and the whole DNA can be synthesized and at the end sealed by the ligase. So nucleotide excision repair has 4 important proteins: UVRA, UVRB, UVRC, and UVRD. UVRA is to scan for distortions within the DNA, the distortions that are because of any problems with the bases. Second thing, UVRB is the one that melts the DNA at the site when a distortion has been detected. And then UVRC is the enzyme endonuclease which makes nicks on 5' side and 3' side of the distortion. And then UVRD, which is the helicase which eliminates the nucleotide, that is, which has the problematic base. And then filling up is done by, as usual, DNA polymerase and ligase. This nucleotide excision repair is also involved in what is known as transcription coupled repair. Say if RNA polymerase is the one that is going to transcribe, and if RNA polymerase is transcribing, and if there is a base problem there, RNA polymerase cannot go further. So it halts there. anytime these kinds of RNA polymerase pausing happens, UVR-AB are recruited there, which would means if RNA polymerase is pausing, which would indicate that there is a problem, and that is why RNA polymerase is unable to move further. And that will allow the recruitment of UVR-AB, and the nucleotide excision repair will take over and repair the damage, and so that in the next round of transcription, RNA polymerase can do it better. I hope that is clear. Thank you very much. See you later.